Can CRISPR make my children taller, more beautiful, and smarter? The idea of using gene editing technologies like CRISPR to enhance human traits such as height, beauty, and intelligence is both fascinating and controversial. CRISPR, short for clustered regularly interspaced. Short palindromic repeats is a powerful tool that allows scientists to make precise changes to DNA. While CRISPR holds immense promise for curing genetic diseases, using it for human enhancement raises numerous ethical and scientific questions. The science behind trait enhancement. Height. Height is a polygenic trait, meaning it is influenced by many different genes. While some genes have been linked to height, it is the complex interaction between these genes and environmental factors that ultimately determines how tall a person will be. In theory, CRISPR could be used to edit some of these genes, but the outcomes are unpredictable. Additionally, the ethical implications of editing multiple genes to increase height are significant. Beauty. Beauty is subjective and culturally defined, but certain physical traits are often considered universally attractive, such as facial symmetry and clear skin. These traits can be influenced by genetics, but like height, they are the result of multiple genes and their interactions with the environment. Using CRISPR to enhance beauty would involve editing a complex network of genes, which could have unforeseen consequences. Intelligence is one of the most complex human traits, influenced by a combination of genetic factors and environmental influences, such as education and upbringing. While some genes associated with cognitive abilities have been identified, the genetic basis of intelligence is not fully understood. Editing genes to enhance intelligence would be highly speculative and ethically contentious. CRISPR, the revolutionary gene editing tool. Imagine being able to rewrite the instructions that make you who you are. That's the power of CRISPR, a groundbreaking gene editing technology that is transforming science and medicine. If you're looking to understand CRISPR, think of it as a high-precision tool that allows scientists to make very specific changes to DNA, the blueprint of life. Here's a detailed look at what CRISPR is, how it works, and its incredible potential. What is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a natural system found in bacteria that acts as an immune defense mechanism against viruses. When a virus attacks a bacterium, the bacterium can capture snippets of the virus's DNA and store them in its own genome in a CRISPR array. This allows the bacterium to remember the virus. If the virus attacks again, the bacterium can produce RNA segments from the CRISPR array to target and cut the viral DNA, disabling the virus. Scientists have harnessed this natural system to create a powerful gene editing tool. By guiding the CRISPR system to specific locations in an organism's genome, scientists can cut DNA at precise spots and make targeted changes. How does CRISPR work? CRISPR technology involves two main components. One, guide RNA, gRNA. This RNA molecule is designed to match a specific sequence of DNA. It acts as a guide, directing the CRISPR machinery to the exact location in the genome that needs to be edited. 2. Cas9 protein. Cas9 is an enzyme that acts like molecular scissors. Once the gRNA guides Cas9 to the target DNA sequence, Cas9 makes a cut in the DNA. Here's a step-by-step breakdown of the CRISPR process. First, designing the gRNA. Scientists create a gRNA that matches the DNA sequence they want to edit. The gRNA is designed to be complementary to the target DNA sequence, ensuring it binds precisely. Second, introducing CRISPR into cells. The gRNA and Cas9 are introduced into the cells that need editing. This can be done using various methods, such as viral vectors or direct injection. Third, binding and cutting. The gRNA guides Cas9 to the target DNA sequence. Cas9 then cuts the DNA at this specific site. Finally, DNA repair. Once the DNA is cut, the cell's natural repair mechanisms kick in. Scientists can harness these repair processes to make precise changes. Non-homologous end joining, NHEJ. This repair method often introduces small insertions or deletions, which can disrupt gene function. Homology-directed repair, HDR. This more precise repair method can be used to insert new DNA sequences at the cut site, allowing for precise gene editing. Applications of CRISPR. CRISPR's ability to make precise changes to DNA has enormous potential in various fields, especially in medicine. CRISPR can be used to correct genetic mutations that cause diseases. For example, sickle cell anemia. By editing the defective hemoglobin gene, CRISPR can potentially cure this painful and debilitating condition. Cystic fibrosis. CRISPR can repair mutations in the CFTR gene, offering hope for a permanent cure for this chronic disease. In research, CRISPR allows scientists to study the function of specific genes by knocking them out or modifying them. This helps in understanding diseases and developing new treatments. The future of CRISPR. The potential of CRISPR is just beginning to be realized. Here are some exciting possibilities on the horizon. Curing genetic diseases. Clinical trials are already underway to use CRISPR for treating genetic diseases. As techniques improve, more conditions may become treatable. Personalized medicine. CRISPR could enable tailored treatments based on an individual's genetic makeup, leading to more effective and targeted therapies. To make our children smarter or more beautiful, let them decide how they want their future.